the last class we discussed about phycomycetes. The different characteristic feature of the phycomycetes is whatever the mycelium is there. The mycelium majorly having the xenocytic condition. Means whatever the mycelium is there, within that mycelium septas are absent. If the septas are absent, there is a nucleus are present, right? So many number of nucleus are present. Many number of nucleus are present. So within the mycelium, without septas, many number of nucleus are present. The condition is called as xenocytic condition. That is about the phycomycetes, one of the different characteristic features, right? So we discussed there is an saprophytic fungi, right? And some are parasitic fungi are there, right? Some are grown on the tongue, some are grown on the tongue. Remember, those the fungi which are grown on the tongue, the fungi are called as coprophilous fungi. The fungi which are grown on the tongue. The fungi which are grown on the tongue. Okay, that fungi are called as coprophilous fungi. Right? That is about the phycomycetes. Coming to today's class, we are going to discuss about ascomycetes. Within the ascomycetes, fungi, the very first one. Other name for the ascomycetes is the sac fungi. We know the phycomycetes. The phycomycetes, other name is algal fungi, because mostly grown on the thallus, algae. Algae having the body structure is called as thallus, undifferentiated cell mass. Especially there is no root and no stem and no leaf-like structures. So upon that the fungi is grown. That's the reason why phycomycetes is called as algal fungi. Here ascomycetes is called as sac fungi. So why here ascomycetes is the sac fungi? Very simple answer. Here ascomycetes, whatever that sexual reproductive organs are there, sexual reproductive organs forms the sac-like structures. The sexual reproductive organs forms the sac-like structures. That's the reason why the ascomycetes is called as sac fungi. The ascomycetes is called as sac fungi. And the next point is the habitat which are grown on wet soils, especially within the wet soils, the fungi is grown, and these are decomposers. Majority of these are decomposers. See, involved in the decomposition, whatever the dead, decaying, maybe the plant parts, maybe the animal parts, right? See, which are involved in the decomposition process, right? That is about that habitat. And coming to the characteristic features of the mycelium, septated branch. Here septas are present and branches are present. And next one is dikaryotic condition or monokaryotic condition. Whatever the mycelium is there, within that mycelium septas are present. So this one is septated. Right? Here two nucleus are present, that one is dikaryotic. Only one nucleus is present, that one is called as monokaryotic. So here these fungi majorly having the dikaryotic condition. In certain cases monokaryotic condition is also present. In certain cases monokaryotic condition is also present, right? So that is about that the fungi, right? Dikaryotic condition and the monokaryotic condition. And coming to the classification based on the structure, ascomycetes can be separated into two groups. One is unicellular fungi, other one is the multicellular fungi. We know the difference between unicellular organisms and multicellular organisms. This is a fungi, yeast. This is that one is only having a single cell. So that kind of fungi are called as unicellular fungi. And see, this is the fungi, right? Here, many number of cells-like structures are present, or cells are present. This condition is called as multicellular condition. This condition is called as multicellular condition. So, ascomycetes, unicellular fungi is there. Here, example is the yeast. Saccharomyces cerevisiae. Yeast is the best example. Okay, other name for the yeast. The scientific term for the yeast is Saccharomyces cerevisiae. Remember, Saccharomyces cerevisiae is the unicellular fungi. And come to the next one, multicellular fungi. Best example, penicillium. We know about the penicillium, right? Other species also there. Okay, Penicillium rotatum. One of the species is Penicillium rotatum. Another one is Penicillium chrysogenum. Another one is the Penicillium chrysogenum. Right? So Penicillium rotatum, Penicillium chrysogenum. Right? These are the penicillium species which are responsible for the production of penicillin. Which are responsible for the production of the penicillin. Penicillin is the antibiotic. Penicillin is the antibiotic. During the Second World War time, Alexander Fleming, produced, that is nothing but the production of penicillin. Production of penicillin done by the Alexander Fleming. He is one of the best scientists. He got the Nobel Prize, right? And during World War time, you see the miracle medicine. You see the penicillin is the miracle medicine because whatever the during the World War time, some of the soldiers are wounded. The soldiers having wounds, the wounds are treated with the antibiotic penicillin, and there is an immediate recovery noticed by some of the persons, some of the doctors. So finally, what I would like to say here, penicillin first, identified or produced by Alexander Fleming. Alexander Fleming. He got the Nobel Prize. Okay. And coming to the next one. Claviceps. This is the one of the fungi. This causes the disease, ergot disease. Right? Ergotin is the one of the compound. Ergotin is the compound present within the claviceps. Causes the disease is the ergot disease. Remember neurospora. This is a very important one. Neurospora. This is one of the best source for the study about genetical diversity. This is one of the best source to study, especially genetical diversity. Genetical diversity studied within which fungi? Neurospora. Breeding and turtle. Both the scientists also best scientists. They propose one of the theory. One gene, one engine. Right? Especially research did on the neurospora. See, I like to do one statement regarding the neurospora. Whatever the genetical variations are there, or genetical diversity is there, genetical diversity related research completely depends or completely done on neurospora. And one of the best example, morels. Morels are there, and next is the truffles. Morels and truffles. These are edible fungi. And these are most costliest fungi. Most costliest fungi. Remember, morels and truffles. These are the edible fungi, and these are costly fungi. Right? That is about some of the gastric features. And see, coming to the next one is the sexual reproduction process. See, within the asexual reproduction process, there is a conidiospores are produced. Conidiospores. The spores are called as conidiospores. I like to discuss about here. This is the one of the penicillium structure, right? Or penicillium fungi. See here, there is a asexual spores are produced. Say this is a reproductive part, and this part is, okay, the part is connected to the one of the mycelium. This particular part is called as conidiophore. The particular part is called as conidiophore, right? The conidiophore contains the conidia. Conidiophore contains the conidia, right? Conidia contains the conidiospores. Conidiophore, TH1, right? Connected to the conidia. Conidia is connected to conidiospores. Conidiospores, right? So here, here example is the penicillium. Here example is the penicillium or aspergillus. Aspergillus niger. Aspergillus niger, the complete name is. This is the fungi name, Aspergillus niger, right? See, penicillium and Aspergillus niger produces the conidiospores. Sir, what is, okay, why is it called as conidiospores? Remember, these are the asexual reproductive organs and these are exospores. The conidiospores are exospores. The conidiospores are exospores, right? So the conidiospores are produced by the mitosis process. The conidiospores are produced by the mitosis process. By the mitosis process, asexual spores are produced, especially within the penicillium and the aspergillus. Okay, then there is a conidiophore is there. That conidiophore is connected to conidia. Conidia having the conidiospores. Conidiospores are non-motile spores, and these are exospores. These are exospores because these are not covered by any membrane. These are not covered by any membrane. Right? That is about the asexual reproduction. And next one we will discuss about the sexual reproduction. Yeah, coming to the next one is the sexual reproduction process. We know within this within fungi the sexual reproduction process, whatever that myceliums are there, means here the two different myceliums are there. Both the myceliums are attracted. One is considered as plus strain, other one is considered as minus strain. So here the plus strain and the minus strain, between these two there is an attraction is there. Because of that attraction, these are arranged very closely, right? After that arrangement process, whatever that cell walls are joined, the cell wall lesions are, especially that cell wall is completely breaking down, right, by the secretion of chitinase enzyme. Once that part is hydrolyzed or broken down, the two cytoplasms are fused. So fusion of the protoplast or the fusion of that cytoplasm, especially the struct is called as plasmogamy. The struct is called as plasmogamy. Fusion of two cells protoplasms, the struct is called as plasmogamy. After that, next step is the fusion of two nucleus. Okay, actually nucleus is nothing but carrier indicates here nucleus, right? See, the two nucleus are fused. The condition is called as karyogamy. After the fusion, see, generally the fungi body state is the end state. The fungi body state is the end state. Once the two nucleus are fused, the state is the two-end state. The state is the two-end state, right? So here the zygote formation takes place. That zygote undergoes meiosis process. Once the meiosis takes place, the two-end state body is converted into the end state. The two-end state body is converted into the end state. So here the zygote having the two-end state involved in the meiosis process produces the four spores. These are sexual spores, right? And see, actually these spores are also called zygospores. These spores are also called zygospores because producing from the zygote, and here four spores are produced. right? Generally we know the concept of the mitosis and the meiosis. Within the meiosis, okay, 4 daughter cells are produced. Within the mitosis, 2 daughter cells are produced, right? So here one cell, okay, involved in the meiosis, 4 spores are produced. Again, all these 4 spores are involved in the mitosis process. We know the concept, one cell in mitosis produces 2 cells. So here, totally 4 cells are there, or 4 spores are there. These 4 involved in the mitosis, right? Finally produces the 8 spores. So in a ascus structure, how many number of spores are present? 8 spores are present. Completely matured ascus contains the number of spores are 8. See, there is different types of ascocarps are there. See, ascocarps are nothing but whatever the ascus is there. Okay, the ascus is covered by the mycelium and forms the different types of structures based on that. Okay, these are called as types of ascospores, and these are also called types of fruiting bodies. So finally here the fruiting bodies are produced. The very first one is called as clistotecium. Clisto indicates here closed. Right? So the ascocarp structure is completely closed, and the shape is round shape or ball shape. The shape is round shape or the ball shape, right? So here closed and round shape or the ball shape. See, this is a closed structure, no opening. Okay, in the middle region, the ascus is present. So here example is the vijija. Right? And coming to the narrow opening is present, especially this is the mycelium. There is a very narrow opening is present, right? See, the shape is the flask shape. This is called as perithecium. Perithecium flask shape, right? So here example is the penicillium. Okay, P, perithecium, flask shape, and penicillium is the example, right? And last one is the apothecium. Okay, the structure having the broad opening. Especially, okay, this one forms the saucer-shaped structure. Right? Example is the clavicles. So this is about the different types of ascocarps which are involved in the sexual reproduction process. Right? And I like to give one of the best example. I already said Aspergillus niger is one of the fungi. That fungi produces citric acid. Citric acid is very, very useful. Okay, within different types of food industries. We are eating candies. Okay, we are eating some other chocolates. Okay, different types of chocolates are there. Okay, see, all these chocolates majorly mixed with the citric acid. Because the citric acid, okay, inhibits the growth of the microorganisms. The citric acid inhibits the growth of the microorganisms because the presence of citric acid, pH is decreased. Right? So once the pH is decreased, the microbes are unable to grow because pH is not favorable for the growth of microbes. Right? So that is about one of the best example as well as niger. It produces the citric acid. Right? Yeah, this is about sexual reproduction and the types of ascocarps, and these are the examples. Okay. Next class we will discuss about the vesicomycetes.